All right, welcome back to the deep dive. Today, we're going to be taking a pretty fascinating look at how the environment basically acts as this master conductor. It really influences how plants and microbes produce those valuable compounds we hear so much about,
Yeah.
you know, secondary metabolites.
Yeah,
These are really nature's little powerhouses. We're talking about things like the flavors in food, life-saving drugs. I mean, the list goes on and on.
absolutely.
And for this deep dive, we've actually gone and pulled a bunch of different sources that focus on how these organisms change, what they're doing, how they produce these things based on their surroundings.
Yeah.
And the goal today is to really give you, the listener, the main things that you can actually tweak in the environment, kind of the levers you can pull to really boost production of these compounds.
Right, exactly. Instead of going down into like the deep molecular pathways and all that,
Right.
we're focusing on kind of those signals, those stresses that these organisms, they've evolved over millennia to respond to. And we can leverage that to produce more of what we want.
Totally. So let's start off with one that everyone's probably pretty familiar with, which is light.
Okay.
And I think most people kind of realize that it's really vital for life on Earth, but it's more than just energy, isn't it?
Yeah.
I mean, different types of light are actually sending really specific signals to these organisms.
Absolutely. And, what's fascinating to me is how specifically tuned these organisms are to different parts of the light spectrum.
Oh, interesting.
So take, for example, UV light, right? So UVA, UVB.
Okay.
These wavelengths of light often will actually trigger the production of these protective compounds like flavonoids, phenolics, things like that.
And so it's not just, I guess, about knowing this just for the sake of knowing it, but if we know this, we can then actually use this information to, let's say, expose plants to very specific types of light.
Exactly.
Maybe we can even increase the nutritional or the medicinal value of these plants by boosting those compounds specifically.
Right.
So it's not just the type of light, but also how intense the light is, right?
Yeah.
And too much light, can that actually be a good kind of stress?
Well, yeah, it definitely can be. When a plant is, senses high light intensity, sometimes it can lead to what we call oxidative stress.
Okay.
But to deal with that, they often will increase their production of antioxidants.
Okay.
So in that sense, we can use that, in a controlled environment to actually increase, the antioxidant content of some of these plants and even the length of the day, the photo period.
Oh, wow.
That can have an impact on these stress responses and ultimately how much, let's say, phenolics are accumulating.
Okay, so we've talked about light and how it can trigger some of these protective mechanisms.
Yeah.
Now let's move on to another one that I think everyone listening is familiar with, which is temperature.
Uh-huh.
We know there's an ideal temperature range for most things to grow,
Right.
but what happens when it gets too hot or too cold outside of that comfort zone?
Right. Well, if we think about this in terms of survival, right, going back to that,
Yeah.
temperatures outside of that range, let's say, a lot of plant cultures, 20 to 30 degrees Celsius, that's kind of the sweet spot.
Yeah.
But if you go outside that range, either direction, it can actually induce some stress.
Okay.
And what's interesting is that stress, a lot of times it can lead to the production of some of these protective secondary metabolites.
Okay.
So things like heat shock proteins, these compounds that help deal with that stress.
So are there researchers looking into ways to maybe, let's say, cycle the temperature up and down
Yeah, absolutely.
to kind of mimic what happens in nature,
Yeah.
like day and night?
Right. And so there's a lot of research going into that.
Okay.
But it's also a delicate balance because you can also go too far, right? You can go to such extremes of temperature that you actually inhibit growth.
Gotcha. So there's a sweet spot we want to be aiming for.
Yeah, exactly.
Okay.
Finding that, Goldilocks zone, so to speak.
Totally.
Yeah.
So let's switch to something else. Let's talk about pH.
Okay.
Now, this is all about acidity or alkalinity, but it has a lot of implications, doesn't it?
Oh, absolutely. The pH of whatever medium these organisms are in, whether it's soil, water, growth media,
Right.
it's really critical because it affects how nutrients are available to these organisms, how they can take those nutrients up, and how the enzymes that actually do all the work of making those secondary metabolites, how well they can function.
Gotcha.
So it's really, that Goldilocks zone again for enzymes. And a lot of times, for a lot of plant cultures, we're looking at around that 5 to 7.0 range.
Okay.
But then there's also the fact that the organism itself, inside its cells, it has to maintain a certain pH as well.
Gotcha.
And if the external pH shifts too much, that can actually really disrupt that delicate balance.
Okay. So it's both the outside and the inside that are important.
Exactly.
All right. Let's move on to water.
Okay. Obviously, we know that water is completely vital for life.
Right.
But too much or too little, that can also create issues.
Yeah.
And one of the big things that kept coming up in the sources was drought stress. What's the typical response that a plant would have to drought conditions?
Well, drought, it's a huge challenge for plants, and it leads to not only osmotic stress but also oxidative stress.
Yeah.
And what we see is that the plants, they'll start producing these osmolytes, things like proline,
Okay.
that help them maintain their cell structure when water's scarce.
Yeah.
But we also see an increase in those, same secondary metabolites that
Yeah.
we've been talking about, the antioxidants, so phenolics, flavonoids, even a hormone called abscisic acid.
Okay.
They're all part of that plant stress response.
No, we often see this term water potential. What does that mean?
Oh, yeah, that's basically a measure of how easy it is for a plant to get water.
Okay.
So when there's a drought, that water potential goes down.
And it makes it harder for the plant to absorb the water it needs.
Makes sense. Now, if we think about growing things in bioreactors, where we might have microbes or even plant cells in this liquid,
Uh-huh.
aeration becomes really important, having enough oxygen.
Yeah, absolutely.
Why is that?
Well, oxygen is actually used directly in a lot of the biochemical pathways that create these compounds.
Wow.
So there's these enzymes called oxygenases that actually take that oxygen and incorporate it right into those molecules.
Okay.
So, when you're growing things in a bioreactor, you really have to make sure there's enough dissolved oxygen in that liquid.
Gotcha.
Otherwise, those organisms, they can't grow as well.
Yeah.
And they can't produce, as much of what we want.
Okay. And actually, too little oxygen can also cause stress, right?
Yeah, that's right. Low oxygen can trigger its own kind of stress response.
Okay.
And it can cause the cells to shift what they're doing metabolically.
Gotcha. So there are a lot of things to consider with oxygen. Okay, so let's talk about the last one here on our list, which is osmotic stress. Now, that sounds kind of similar to drought stress, but I'm guessing it's a little different.
Yeah, it is. You're right. They're related. But while drought is about, how much water is available in the soil, osmotic stress is usually caused by high concentrations of salts, maybe sugars, other solutes in that environment.
Okay.
So it kind of has the same effect on the organism. It's harder for them to get the water they need because of that imbalance in the solutes.
Gotcha.
But the response is often very similar.
Okay.
We see the production of those, compatible solutes that we talked about, and also those stress-related secondary metabolites that help them cope.
All right. So just to do a quick recap, the main environmental factors that we've gone over today are light, and that includes the wavelength, the intensity, how long they're exposed to it, temperature,
Right.
Finding that sweet spot,
Yeah.
making sure we understand those stress responses, pH, both inside and outside the cell.
Absolutely.
Water availability,
Right.
oxygen levels, particularly for things that are growing in those bioreactors.
Exactly.
And then osmotic stress. And I think what's really cool about all of these is that, these organisms, they don't just sit there passively. They actually respond to these signals and these stresses by changing what they produce chemically.
Exactly. And it's really that responsiveness that we can leverage, right? So in a controlled environment, if we can really dial in these factors, we can fine tune and really get the most out of those systems.
Yeah, it's almost like, winemakers. When they carefully control the oxygen during the fermentation process, it has a huge impact on the final flavor.
Right.
And I think here it's kind of the same concept, just on this tiny biochemical level.
Yeah, absolutely. It's all about understanding those, those subtle cues and how they can have a big impact.
Totally. Well, this deep dive has really just been so fascinating to see how sensitive these biological systems are to the environment. And I think it makes you wonder, considering how intricate all of this is, all these little interactions, what other subtle environmental cues are out there that we might be missing?
Right, exactly. What else could we be tweaking that we haven't even thought of yet that could, really push the production of these compounds even further?
Absolutely. Well, thank you so much for joining me on this deep dive.
It was a pleasure.
And for all of you listening, if you have any thoughts or ideas, make sure to leave a comment below. And we'll see you next time.
See you then.